Okay, this is Chapter 12, DNA Technology and Genomics, Part 2. All right, restriction fragment length polymorphism can be used to detect differences in DNA sequence. And all that means is that when we chop up the DNA, it's going to be in different lengths for different people, and we can figure out the genes they have by the length of those fragments. So let's take a look at that. All right, so you have restriction, fragment, reflect, DNA sequence, right? Remember, the restriction enzyme cuts it. So when we look at where these things get cut, it's going to tell us something about the DNA. So in this slide, you have two pieces of DNA. And you have a restriction enzyme that's going to cut between the CCGG little section of DNA here. You can see it right here. This is where this restriction enzyme cuts. It's always going to cut that code. So when we look at two different people's DNA, that code might show up in different spots depending on the individual. So on this individual, this is the DNA taken from the crime scene. In this individual, this restriction enzyme is going to cut here, and it's going to cut here. That's two cuts. And when you do two cuts, you're going to end up with three pieces. So you're going to have sort of a kind of medium small piece here that you've cut. You're going to have a longer piece here in the middle you've cut, and a really pretty small piece here that's been cut. The suspect that they're looking at, same restriction enzyme, only cuts this DNA in one spot right here. Okay, so we end up with a rather very long piece right here and a very short piece. And those are going to look different when we do the gel electrophoresis. The length of these are going to be two different things. All right, so we've cut up that DNA, and you now run it on the gel electrophoresis. And as you recall, that the longer fragments don't move as far. So the one that was cut in three spots, all those fragments are going to be a little bit shorter. You can see here that they went a little bit farther because they're a little bit shorter. The suspect who only had it cut in two spots had a very long fragment here and then the very short fragment right there. So one thing we can do with DNA is we can test for harmful alleles, things like Huntington's disease or cystic fibrosis. We can look for the alleles of the gene that would code for Huntington's, and we can do that using the radioactive probes similar to what we saw before. So how do you do that? Well, it's very similar to what we were doing before. You need to have an example of somebody with the disease. So maybe right here, person 1 has the disease itself, because you'll need that to compare to see if you have a healthy person. And then use the restriction enzyme. You cut up the fragment, same as always. You run it. And then when you run it, you don't want to tag every single DNA sequence. You're looking for certain ones. So you can use these radioactive single-strand probes, the ones that are going to hunt out for certain fragments here. And you can kind of wash the gel with these and then rinse them off. And they're only going to stick to certain ones that are complementary to the probe. So you can see here, and I don't remember who they think has it, but let's say person 1 right here has the disease. Well, then person 2 would also have the disease, but not person 3, because it's cutting it in different lengths. All right? DNA technology is now commonly used in courts of law. That, like I said earlier, that didn't used to be the case. It is very, very common now. Okay? How do we do that? Well, we can do DNA fingerprinting to help solve crimes. We take a few different markers that they look for, a couple different alleles, and see if the defendant and the victim or the suspect, whoever they're looking at, they can see if those markers match. Gene therapy may someday be able to help treat a variety of diseases. Now, the hard part of gene therapy, of course, is getting that gene into every single cell in an adult body when we look at the way adults grow, when you're first an embryo or a single cell that's been fertilized, that DNA goes into every single cell. But if you were to just take DNA and insert it into a few cells, it's not going to make it into every single cell. They've tried for people who have immune suppressed, a genetic immune, a genetically suppressed immune system. They've tried injecting it into the bone marrow, which is like the stem cell area. From the bone marrow, all the white blood cells will be made. They did that on some kids and found the gene that they injected was actually really close to a leukemia gene, and they switched that gene on by mistake. So there are risks involved. It's not necessarily a sure thing, but it's definitely an interesting way of going to think of getting a gene into every single cell and then being able to fix the problem. Um, we would like to think one day we could use it to treat both genetic diseases and non-genetic disorders, but like I said earlier, it's difficult to get the DNA into every single cell of your body. All right, the PCR method is used to amplify DNA. And it's called polymerase chain reaction, and it can be used to clone a small sample of DNA quickly, producing enough copies for analysis. So if you're at your crime scene and there's like one tiny drop of blood or a splatter or something, and you don't get enough DNA to actually do your analysis, you can do this PCR, and that will give you enough DNA that you can run your gel electrophoresis and all the other things we do with DNA. And it's a very simple idea of just taking your polymerase and having it clone DNA. The cool part of it was they found this enzyme in a very heat-resistant, I believe it was a bacteria, heat-resistant bacteria, because part of the process of this is heating and cooling your DNA over and over and over again to cause it to replicate. And not all polymerases were heat tolerant like that. So that was a cool invention there. The Human Genome Project has been going on. They've completed, last I checked, they completed the Human Genome Project so far as mapping of the human genes. I think when they started in 90, they thought it was going to be done like in 15 years or something, and it's done. I think they even finished early. And again, the idea here is if we have the genes, we can figure out the genetic problems. Okay, so the data is useful both for genetic disease, but also looking at evolution. If you want to look at not only the evolution of humans, like how do we compare to other animals, but also human evolution. It gives you an insight into all different kinds of things. There's now evidence that there was mating between Homo sapiens and Neanderthals. And so there's a certain amount of genes from Neanderthals. So anyway, it provides all kinds of interesting stuff. Okay, most of the human genome does not consist of genes. And remember, a gene is a section that codes for a protein. There's only about 25,000 genes. And forever people thought, well, it's got to be hundreds of thousands because, what's so important, proteins. But it's only about 25,000. And for a long time, they were calling the other stuff, well, junk DNA. Well, it must not do anything. But it turns out that it does stuff. Some of it we still don't know, but some of it's structural, holding chromosomes together. Some of it's protective, and some of it's regulatory, helping turn genes on and off. Much of the non-coding DNA consists of a repetitive nucleotide sequence. And where we think that might have come from are these things called transposons. And they can actually move about and copy stuff in the genome. They can jump around. You'll find a lot of this repetitive nucleotide sequences by the centromeres and then by the ends. So there's probably a structural component to it. But we think it got there by these transposons that actually jump around. All right, then the science of genomics also can compare whole genomes. So there's many different, we've sequenced both prokaryotic and eukaryotic genomes, and that produces a bunch of data for the study. So we've not only done the human genome, which is obviously very interesting to us, but we've done things like E. coli or rice or different genomes, so we have a basis to compare. So here you go. Here's some non-human genomes that we can compare. We have done things like Saccharomyces cerevisiae yeast, very important in the laboratory. Also good for making beer and wine, but also used in the lab. E. coli, hugely used in the lab. C. elegans, which is a kind of nematode, again, very useful in the lab, so that's probably why all these are getting their genomes done. Fruit fly, very commonly used in the lab. Mustard plant, again, commonly used in the lab, so is a mouse. What's interesting is rice is not that commonly used in a lab, but it was one of the first genetically modified organisms out there, one of the first GMOs. And they actually put a vitamin A in it. A lot of third world countries are low in the beta carotene that makes the vitamin A. And so by studying rice, which is a very important crop for humans, maybe not in the United States, but worldwide, this is a very important crop, they were able to add that vitamin to it. All right. Proteomics is the study of full sets of proteins produced by organisms. And maybe you remember a long time ago in regular bio, I talked about the protonist versus the geneticist who wanted, the protonist thought that the proteins were the heritable factor because, wow, proteins are so cool. Well, proteomics is a little different than that. It's the idea that the proteins do everything and we need to study them. So to get a full set of all the proteins used by an organism is the proteomics. All right, GMOs, genetically modified organisms, are used widely in agriculture. I think 90% of the soybean oil you use is GMO. Some for good and some for bad. Okay, how do we get GMOs? Well, we take our plasmid, we take a restriction enzyme. I'm sure you're used to this idea by now. But we're going to cut out the plasmid and we're going to put it into the DNA and then generate a plant using hormones. So the way to get that piece of DNA into every cell of that plant, remember, you can't stick it into a leaf or a root. It won't go all through the plant. You have to start with that cell as if it were an egg or a seed, really, kind of thing. But you can do that on plants using hormones. You can actually grow them and differentiate the cells into leaves or roots just using hormones. So that's how they get that piece of DNA into every single cell of a plant. Okay? Then you have transgenic organisms, those that have both um, uh, their own DNA as well as DNA inserted into them from another animal. And then we have a number of important crops and plants that are genetically modified. I mentioned soybean, the rice with the vitamin. All right, could genetically modified organisms harm human health or the environment? Well, we can look at Monsanto, who created what's called Roundup Ready plants, and instead of reducing the amount of Roundup, which is an herbicide, instead of reducing the amount used, it's actually increased the amount used because you can spray right over your crop and it'll kill the weeds, but it won't kill the crop, which sounds lovely, except that that Roundup Ready gene, remember how genes can jump in certain things? They have found that Roundup Ready gene in some weeds, which means the very expensive Roundup I use would no longer be usable on those kind of weeds, and it would kind of tick me off. So that could be some definitely downside. Plus, they're obviously using more of that now instead of less of it. So there's some upsides, and there's some, as always in life, there's some downsides. There are risks such as ecological damage from genetically modified crops. Again, the genes jump, um, and that can be dangerous. Um, so, yeah, again, ups and downs. There's a genomics researcher, Eric Lander, discussed the Human Genome Project. He was like the genius from MIT. Um, anyway, he points out that the Human Genome Project, even though we're at the end of where we've finished the sequencing, all that information that we can gather from it has yet to be put, kind of put to use yet. They're still working on how it all fits together and how we can better human life using that information. Okay, so an extra slide. We already did that one. Oops.